Okay, so just to give you an overview of what I want to speak about today, I do want to give you a very brief introduction to Nexcelom Bioscience as a company, and then talk about the importance of accurate cell counting specifically for stem cells. I'll then introduce image cytometry as an alternative method for doing stem cell research, and particularly focus on IPSC reprogramming using the Celigo. So we'll go over establishing cell cultures, transfection efficiencies, establishing an expansion of stem cell colonies, and then characterization of those colonies as well. The last couple of applications I did want to cover is differentiation and how you would look at that using imaging. And some people doing stem cell research are also very interested in that coupled with migration and wound healing models. So I just wanted to touch on that at the end as well. And then we should have plenty of time for any questions. Okay, so Nexcelom, as a company, we were founded in 2003 in Boston in the USA. Our headquarters is in this converted red brick mill building that you can see here. That's where all of our manufacturing development goes on, just there. Our European base is in Manchester, that's where I am, and we work really closely with Immuno Diagnostic in Finland as well. We are a company that's completely focused on automated cell counting and image analysis. We don't do anything else. We really are experts in this area, and our instruments are really quality instruments. We're a group of scientists helping other scientists, and we want to increase your accuracy for these kind of really important experiments and save you time when you're running them in high throughput as well. One of our USPs and our company ethos is to provide excellent customer service and application support to all of our customers for the lifetime of all of our instruments. So what do we do? Well, at Nexcelom, we specialize in cell counting, cell assays, and image cytometry, and we have a range of premium top quality cell counters, and that's our Cytometer range, which I'll talk about in a second. The newest addition to the family was launched last year. Oh no, it's about 18 months old now, is the Celigo, which is a high throughput version of these cell counters. I'll also briefly talk about that. And then I'll spend the majority of the presentation talking about how image cytometry using the Celigo can really enhance your stem cell research. Okay, so just as an introduction, the role of stem cells, excuse me, in medicine, and more specifically personalized medicine, seems to be increasing all the time. And laboratory techniques to monitor these cells, these stem cells, at all stages of this process here is absolutely vital. So stem cells, they come in a variety of different morphologies, and of course, once differentiated, they can also vary wildly as well. So you really do need to have good quality analysis systems and counting systems in place to get these accurate results from this kind of research. So I really wanted to emphasize, the importance of accurate cell counting through most of stem cell research. And for most of this process, you will want to be monitoring your concentration of cells, the health of your cells, right from the start, from establishing cells from a patient to characterization, to transfection or gene correction, expanding those cultures or expanding colonies, doing differentiation experiments, all the way through to cryopreservation as well. You'll want to know the health of your cells and if they are being treated nicely. And throughout all of the processes here that I've just mentioned, the cells are going to be changing in terms of their proliferation speed, their morphology, their viability, the size of the cells, maybe clustering factors change, and also if you're transfecting things, the fluorescence may change as well. So it's important to choose a cell counter to monitor cells with all of these different morphologies, sizes, viabilities, different fluorescent potentials that really gives you flexibility in your ability to analyze these cells of all different kinds of morphology, and that will produce consistently accurate results. So at Nexcelom, we really do have quality cell counters, and we don't just have one. It's not one size fits all. We actually have 7 different kinds of cell counters. And some of the cell counters have different fluorescent capabilities, magnifications, etc. But what you can expect from all of them is accurate and precise cell concentration and viability measurements. They've actually been used for over 2,000 cell types now that have been characterized using these cell counters, and we have characteristics saved for all of those 2,000 different cell types that you can use. All of our cell counters offer flexibility in terms of cell size, morphology, and the clustering parameters as well. They have a range of magnifications depending on what cell type you're using, and a range of fluorescent detection modules as well. They have enhanced declustering capabilities. I know from my work with stem cells that a lot of them can be quite clumpy, difficult to work with. The declustering capabilities of our cell counters are absolutely fantastic. And they also, some of them as well, do have the ability to do cell-based assays. So you can do your transfection efficiency and differentiation assays for single samples on these cell counters. And if you do need all of that, but in higher throughput, then the Celigo MX will run 24 samples in a single run with the same degree of accuracy and consistency. So I'm not going to talk about cell counting anymore. I really wanted to move on to image cytometry and the Celigo. But first, I did want to mention some classical methods for producing results from your in vitro stem cell assays. And these are for the more extensive experiments. So some of the more traditional technologies are flow cytometry, using manual microscopy, and using plate readers as well. And all of these are widely used methods, but they do have problems when doing these stem cell assays. So flow cytometers are great for analyzing multiple fluorophores or multiple markers within a single population, but they do require quite large cell numbers and large volumes of cells. They also require trypsinization. So if you've got adherent cells that you really want to keep adherent and image them and look at them in situ, you can't do that on a flow cytometer. The whole process of flow cytometry can take a long time as well if you have a lot of samples, and it's only an endpoint assay. You can't reuse the cells, you can't look at different time points. It's just one single time point at the end. For manual microscopes, you're going to end up with beautiful images, especially if you use confocals, but it's very, very time consuming if you want to take images in multiple channels, overlay those, especially if you have a lot of samples to look at. So it's great for getting really nice images, but for analyzing in high throughput, not so good. And again, there's no analysis software linked to that. You would have to do some external analysis. Plate readers are also fine for getting fast and bulk measurements from things, but you really can't do that many cell-based assays with a plate reader, and sensitivity can be a problem. So with those disadvantages in mind, I just wanted to introduce to you image cytometry. So image cytometry is basically like having a microscope in a box, but with a high-speed microscope, let's say, and with automatic movement from well to well within plates as well, meaning that you can do these kind of experiments in high throughput. Most image cytometry systems, and including the Celigo, also have analysis software built into them. So you can also do the analysis part within the same software and run these assays and get your results in a really short space of time. So just the key points about that: you can image your cells within your plates. There's little maintenance, no contamination problems, and they're easy to use. They really are very fast, and I'll give you some examples of the speed of those in a second. And you still get these multi-channel publication-worthy images on top of all of that. Okay, so I'm just going to focus a little bit on the Celigo technology and just talk a little bit more about that. It has 4 fluorescent channels and a bright field channel to choose from. You can image in all 5 channels at the same time, get beautiful overlaid images. It's compatible with anything from 1536 well plates all the way to 6 well plates, so you can have your cells in any of those, and also with Petri dishes, slides, and tea flasks. And the great thing about this particular image cytometer is it does whole well imaging really well. You might find some other image cytometry systems that will just take an image in the middle of the well as a representative area, and it's not always a good idea to just take an image in the middle. As we know, cells don't necessarily grow homogeneously across wells. They can gather around the edges, they can gather in the middle, and they can grow very heterogeneously. So it's a much better idea to look at the whole well and analyze every cell within that well. So just a little bit more on that point, the whole well imaging. If you are trying to monitor the proliferation of your stem cells, for example, and maybe you're looking at different media compositions to see which media is causing them to proliferate more quickly or to be more healthy, for example. Again, you would really want to look at the whole well for that. We've got an example here of someone who didn't look at the whole well. They just took an image in the center of the well and used that to estimate the confluency of those wells. The yellow fill that you can see here, it's not cells, it's not fluorescence from the cells, it's just indicating where cells are present within that window. So in image A, we can see that it's almost completely covered that window with cells, so it's 95% confluent. In image B, we can see it's only 40% confluent. There's a lot less yellow in that image. But if we reveal the whole picture and we see the whole well image, we can actually see that they're both 70% confluent. It's just due to the heterogeneous distribution of the cells that are causing these differences when you only look in the center. So it's really important to do this whole well imaging. Another feature of the Celigo that I really wanted to mention is the ability to do label-free counting of adherent and suspension cells. Not just label-free counting, but label-free morphology assessment as well. And I'll come on to that a little bit later. But what we've got here is 8 different cell types. You can see that some of them are suspension, some of them are adherent, some of them are quite clumpy, some of them are not, some of them are elongated. But what you hopefully can see here is the green outline of all of those cells. And that is the Celigo software analyzing all of those different cell morphologies equally well and getting you accurate cell counts. And my last point about the Celigo really is you do have flexibility with the fluorescence as well. It means having 4 fluorescent channels means you can look at different stem cell markers, for example. If you want to look at 4 different markers, you can have those in there. This is just an example with 3 different markers where you can see, and we're looking just at viability here, but you can add an extra marker on top of that and utilize these multiple fluorescent channels. I've just got an example here just showing you how the Celigo will outline those different cells with the different markers present. Okay, so moving on to the thing I really wanted to focus on, which is IPSC reprogramming using the Celigo. And here are the key steps in that reprogramming process, all of which the Celigo can help with and help you streamline this process. So first, establishing and expanding your cell cultures from patient biopsies. Then recovery of cells and achieving optimal cell density for reprogramming and also optimal cell viability as well. Transduction or transfection of cells. Daily monitoring of colonies. Identification and isolation of your IPSC colonies. And then expansion of those IPSC colonies. Characterization of them, so using different stem cell markers, and performing experiments for routine quality control of your cell lines. So I wanted just to show you an example. All of these images are from the Celigo, by the way. Everything is taken on the Celigo. All the analysis is done on the Celigo. But I just wanted to give you an example from each of those steps of what you can achieve if you have an image cytometer. So first with the establishing of cell cultures from primary samples, and these could be adherent cells, these could be suspension cells, or these could be clusters of suspension cells as well, and all of which can be imaged really nicely and quantified. So cell cultures are established, they're expanded and banked from patient biopsies, and the confluency application can be used to monitor the growth of these cells. And that's label free. You don't need to label those with anything. And you can look at multiple different time points as well and produce kinetic data. For suspension cells as well, and for a lot of adherent cell types, you can not just look at the confluency of the wells, but you can also determine how many cells are in each well as well. So here we've got an example of some cells growing at quite a low concentration. We've got 8,000 cells in a 96 well plate there, and that's an example of what that looks like. But we can actually go up to 120,000 cells in a well and analyze those accurately and monitor those over time. Okay, so the next step, creating an optimal cell density and an optimal cell health for reprogramming. That can be really important. So the process is that cells are thawed and allowed to recover prior to reprogramming. Cell counts and cell densities can be calculated using either the direct cell counting application, or you can also relate that to confluency as well. And you will have an optimal cell density that's really essential to increase the likelihood of successful reprogramming events. And this can be really critical in reprogramming how densely your cells are concentrated in the well. And on top of that, you can also perform viability experiments in a number of different ways using the Celigo to look at the health of your cells. I've just got one more example of viability here. This is a group who are looking at viability using acridine orange and propyridium iodide. We can see we've got suspension cells here, and what they're looking at, just as an example, is two different media conditions to see which one keeps their cells most healthy. And we can see here the acridine orange, that's the green dye, that's indicating a live cell. The propyridium iodide is the red dye, and that's indicating the dead cells. We've got media condition A on the left and B on the right, and we can see just from the images that media condition A is keeping the cells much more healthy than media condition B. I've just got a little graph on the right there just to show you that. So maybe this group would continue with media condition A moving forward with their experiments. So the next one, transduction or transfection of cells. So this is a lentivirus titration. So cells are commonly transduced or transfected with reprogramming factors linked to a fluorescent reporter. So that, for an example of that, is GFP. As we have the 4 fluorescent channels, it doesn't have to be GFP, it could be RFP or something else. The transduction or transfection efficiency can be calculated, and conditions including the cell density, which we've been measuring before, and the cell health and the media composition, they can all be optimized to produce the best transfection or transduction efficiency. So you might want to try different concentrations of cells, different media compositions, and see which of those produces the best transduction efficiency. You could run that sort of experiment in a 96 well plate. For this particular experiment, what's been done is the reagent was used at different concentrations. So the transfection reagent, and we can see there at 50 nanograms, we're getting a very high level of transfection, and at 6, and obviously at 0, we see nothing, and at 6, we see something in between, and we can plot those on a graph as well to see which is the most optimal to produce the best transfection efficiency. So establishing colonies sometimes takes quite a long time, up to 4 weeks, but we can look in bright field. These aren't the clearest images, but hopefully what you can see when we get to day 22 is we can start to see these dense colonies appearing in that well there. So the colony establishment is monitored daily by the Celigo's whole well bright field imaging function. You don't need to have any dyes on there. You can just image day after day. It takes around 5 minutes to image and analyze a whole 96 well plate and the whole well using this kind of experiment. It's very, very quick to do. And you'll be able to see when your colonies are starting to appear. So when you know you have colonies, you want to know if they are specifically stem cell colonies. So you can stain with various different stem cell markers, and these can be live dyes if you want to as well. And the colonies that are positive for those stem cell markers can be identified and isolated using different techniques. So once those colonies have been isolated, you can then expand those into known stem cell colonies. So the IPSC colonies are transferred into fresh wells. You can expand them for multiple passages quite often to confirm the stable growth of these colonies, stable growth and maintenance. And again, this is all using the Celigo's colony application, which is specific for stem cell colonies. You might at the end want to do some characterization or quality control of your stem cells. So your stem cell lines can be characterized for the expression of pluripotency markers. Following this, cell lines may be banked so you can cryopreserve them or carried forward for different experiments. And just as a final point here, the Celigo allows the user to miniaturize your experiments as well. So you don't need to do these in really big well plates. You can do these in 96 well or 384 well plates, which means you're saving on precious cell types and saving on reagents, stains, etc. So the last couple of things I wanted to talk about, one of them was differentiation. So if you have some cells and you want to add various different components to the media to see if that promotes differentiation, as an example, this is what I've been doing with one group at the moment. They've been looking at some neural stem cells and adding various different compounds that induce sonic hedgehog. And then they'll stain with calponin 1, and that's the red staining that you can see there with Alexaflor 546. They also stain their cells with DAPI, and they're looking for these red cells and how many calponin 1 positive cells are within that population. So again, these are all images that are just taken straight from the Celigo and can be used for publications. This is how the Celigo will do the analysis part of this. So what it will do is it will, first of all, look at all the nuclei that are in your sample. So it will count all of the individual DAPI stained nuclei, give you a number of those. And then what it will do is it will tell you how many of those have this red stain associated with them. So how many of those are calponin 1 positive, and it will give you a percentage of those. For this kind of experiment, for a full 96 well plate, it will take around 8 minutes to do the scanning and the analysis. So again, if you have lots of different plates in high throughput that you want to run, you can do this very, very quickly. What it can also do, because of the really nice bright field images that you can get, it allows you to look at the morphology or the aspect ratio for each cell within that well with no stain at all. So these are live cells, and what the Celigo does is it gives each cell a score based on how round or how elongated those cells are. And this can be an indication of differentiation without using a fluorescent stain. Obviously, it's not as accurate as when you do use a fluorescent stain, but it can give you that indication. So I've got an example here of some cells where we can see a lot of them are stuck down and they're more elongated, but we also have some round cells in there as well. The red segmentation indicates any cell that is not round, that is more elongated, and the blue indicates any of those round cells. And you can get a percentage that are round and a percentage that are elongated, and that gives you some sort of indication about differentiation. And my final application that I just wanted to cover here as well, if you're looking at stem cells, you might be looking at applications such as migration and wound healing as well. So what image cytometry can do for you is it means you can do things like scratch assays, and you can monitor the cells healing that wound after a scratch assay. So that's done quite simply. You get a monolayer of cells in your well, you produce a scratch down the center, and then again monitor on a daily basis to see that scratch, how that is healing. Another method is to use plates with a plug in the middle, such as these Oris plates. You can seed your cells around the plug. You'll have a hole in the middle, you remove the plug, and then you see how that hole is filled over time. And again, you could use different compounds from well to well to see which compounds help promote that healing process. So this is just an example of someone using one of those plugs. And at time point 0, when they remove the plug, we can see we have a very small ring around the area here. That's in red, what we can see. There's no fluorescence there. That's just indicating the area of interest. And then we can see after 24 hours, 43 hours, 71 hours, how that hole is being filled. And all of that is plotted within the software as well. There's no external analysis here. It's all plotted within the software. And this is what the wound closure would look like. So cells are seeded till there's a confluent well, a scratch is produced. So at time point 0, the scratch looks like this on the left. And then at 24 hours and 31 hours, we can see how that scratch is healing. So just to summarize what I've talked about today, really, how image cytometry can change stem cell research workflow. Well, it will mean that you get your results earlier and finish your projects faster. It can give you more data, more replicas, and if you use smaller plates, you can use less reagents, less cells, especially if they're precious cell samples. You eliminate user variability because everybody's running the same experiments with the same settings. You can also really create really nice multi-channel whole well images that can be used for publications. And all of that is automatically digitally stored as well. So I've covered, this is the Celigo wheel. It is a representation of all the different assays that the Celigo is being used for all over the world. I've covered a lot of the IPSC reprogramming section on the right there, but you can see there's a lot of different applications that it can be used for. If anything else is of interest to anyone, please feel free to get in touch with any of us and we can talk a little bit more about some of those other applications as well. And I just wanted to thank everybody for attending today. We're really happy to discuss anything presented. And if you have a troublesome experiment that you would like to discuss with us as well, we can also help you in ways to streamline those experiments. Thanks very much.